Dear students, in this unit we will talk about the test tube cloning of plants. This is also one of the methods of vegetative propagation of the plants, which means that no sexual reproduction is involved and we get a lot of plant species or the new plantlets in a very short period of time. Now, tissue culture is actually an in vitro aseptic culture of the cells, tissues, or organs or the whole plant under controlled nutritional and environmental condition, often to produce the clone of the plants. Now, when we talk about the in vitro mean outside the body of a plant. So, a small portion of that plant is taken outside into a glass tube and is propagated and is grown in an aseptic environment. Aseptic environment in which there is no fungal or bacterial or viral contamination is involved. So, as a result of this, clone, a true to type of selected genotypes can be obtained in large number. Now, aseptic method of clonal propagation is known as micropropagation. This offers advantage of producing a large number of plantlets in relatively shorter period of time, and space is also very less space is required. So in a very small lab, you can grow thousands of these plants through micro propagations. This is the only commercially viable method of clonal propagation of most of the horticultural crops, such as different orchards with the beautiful flowers. Now, the different stages in the micropropagation that includes preparative stage, then initiation of the culture, then we have multiplication, then rooting of the shoots and transplantation into a new place. So these in vitro cultures can be stored for a longer period of time through cryopreservation. So you can store it for a longer period of time, and whenever you want, you can take them out and you can grow them back. This way, the breeding cycle can also be shortened through the micro propagation. Now, as you can see here in this slide, we can see that test tube contains a very small part of a certain plants. It has developed, as you can see, shoots, and ultimately a time comes when it also develops roots. So they are developed in a small test tube, and it has become very successful when specifically we need to grow horticultural plants. This method is created from just tiny leaf bud or maybe root segment of the mother plants, and this is actually a standard for commercial nurseries and many commercial scale cultivators. They are using this technique to propagate and to produce large number of plants that not only includes potatoes but many flowering plants as well as many fruit trees. Now, through micropropagation, particular variety may be produced in large quantities and the time is significantly reduced. And this method can produce plants free of diseases, because disease-free varieties are obtained through this technique because it is largely aseptic, and this aseptic technique is advantageous in many ways, including this disease-free cultivars, as well as the very short period of time, as well as the large number of plants can be stored in a very small place. So overall, this test tube cloning of plants, it's an advanced technique and it's becoming very useful in order to grow and preserve important plants.